Okay, let's get going. Welcome. Thank you for coming to the workshop. This is a workshop organized by MetabolomicsHub, which you'll learn about today. And the focus of this workshop is cross-repository discoverability, harmonization, and reanalysis of public metabolomics data. So this really is a joint effort. This is a joint effort between EMBL-EBI and Metabolites at EMBL-EBI. This is a joint effort between Metabolomics Workbench at UCSD, along with GMPS Massive, and the BioHub, supported by the Chan Zuckerberg Initiative. And it's actually quite a good time for metabolomics data. Public data is increasing. Metabolomics and other, omics-related omics are being acknowledged as invaluable tools. There's a rapid growth in public data worldwide. Funders and journals are increasingly mandating this, or at least encouraging data sharing in the community. And now there's demonstrated value in data reuse and reanalysis. So, we see these tools such as MASST, PredictRet, new databases coming online, DB, the correlation database for exposomics. But really, there's still challenges in the field related to different formats of public data, different requirements across the repositories, people using different harmonization, different identifiers. But there has been some success in the harmonization of public metabolomics data, such as panReDU, and we think that there's really endless future potential for public data, especially as we come into the era of machine learning, or AI, or building knowledge graphs as well on top of public data. So, based on this, there was a consortium through the main big data repositories. And the idea really is just to start to implement standard data submission and data dissemination practices between the main metabolomics repositories. So we call it MetabolomicsHub. And what we really want to do here is we want to increase the data sets in the public domain. We want to enable data reuse to maximize the value of all the data generated by the community. We want to establish a common voice for engagement, with journals, funders, but also external initiatives. And we want to, the community want to align with proven initiatives that the other domains do. So such as in the genomics world, there's the INSDC, and in the proteomics world, there's the Proteome Exchange. So throughout the workshop, you'll learn a little bit about all the activities we've been doing for the last year. We started about a year ago, or 18 months ago now, and we're just over halfway through. So what we've been doing? We've been establishing the framework for the MetabolomicsHub. So there'll be a common data model you'll learn about. There'll be submission guidelines that the repositories will comply to. And there's a collaborative agreement to discuss issues such as governance. We've been very much working on the infrastructure for this. So, this is a global infrastructure. This is shared across the main repositories. So we've been defining the data workflow, all the utilities, all the libraries that are needed for this data workflow, the APIs, the database. We've been developing a portal that will host all the data sets across the community and across the repositories, and then obviously working with the repositories to make sure that they can integrate with the framework. The other part of the consortium we want to do is there's lots of good community activities out there, so we really want to support this. So we're looking to develop and help adopt and help increase adoption of the open data standards. So there's initiatives such as MTTabM that you'll learn about, the USI, the Universal Spectral Identifier, and there's others that we want to support as a consortium. One thing is we want to make spectral libraries fair across the consortium. So really, there's no place to share your spectral libraries at the moment. You can go and share individual spectra through the spectral libraries, such as MONA and MassBank. But really, to give a spectral library, we want to give an identifier, and we want to give credit to you guys for generating these libraries, for making them open, and for people and to build upon. So here we're going to have a data model for this, again, infrastructure guidelines, and then we're going to reach out for some key demonstrator projects. And then enable these libraries to be integrated across the ecosystem, for example, with GMPS and others. And then the last thing is management. So we really want to engage with the community. We've had some meetings where we go out and ask people, what do you want from this new consortium? How can we enable people to share data? How can we show the value of public data? How can we give credit to the good data generators? So we've had workshops, outreach, and collaborations as well. So today, hopefully, what we're going to do is we're going to highlight the challenges and opportunities in cross-repository discoverability, harmonization and analysis of public metabolomics data. Hopefully we're going to demonstrate some key development, resources, and real-world use cases that span data discovery, harmonized access, and integrative analysis across studies, but also actually across omics layers as well. And then also what we'll do is we'll hopefully promote community adoption of best practices and encourage contribution to shared standards, shared infrastructure, and shared outreach efforts. What do we hope you guys learn or take away from today? Well, we hope that you can understand the current landscape of metabolomics data repositories and the value of public, but also fair metabolomics data, and specifically at scale now. And we hope that we can show you that there are key initiatives and workflows that enable now cross-discoverability, harmonization analysis of public metabolomics data, and we'll highlight some real key some use cases, real-world use cases for this. And hopefully we can show you that there's really value to engage in best practices and community efforts aimed to support this initiative, and also, fair data across the community in general. So this is the agenda for today. We'll start off with Juan Antonio to give us an introduction to Metabolomics, into the MetabolomicsHub from EMBL-EBI. Then we'll have Eoin Fahy from the University of San Diego to talk about standardization of chemical identifiers and what this means for the MetabolomicsHub and the metabolomics community in general. We hope to have Yasin here, but Yasin's flight was delayed, cancelled, so he's flying today. So I will try to cover structureMASST, but if not, he has a talk on Tuesday, so I will direct any questions to him on Tuesday, and you can go and ask him there. Then Juan Antonio will take us through data standards. So, what are the community doing around data standards, and how can we help support and drive adoption? And then we'll show you what we've been working on for the last 6 months, and a data portal that kind of integrates all the old data out there at the moment across all the repositories. So we'll have a demonstration about that, and then we'll open up for a panel discussion, really to get requirements of how this initiative can evolve, what we're missing, and the future of it towards that. So yeah, that's it. Thanks.
Okay, so yeah, good morning everyone. Buenos días a todos. It's really a pleasure to be here in Buenos Aires. So I'm gonna kind of introduce the main activities that we are doing in the context of the MetabolomicsHub consortium. Please, we are gonna be here the whole week. If you have any questions, also for the local people, if your English is not that good, you can still approach me, so, and others as well. And yes, let's start. So this is an overview. I will give you a short introduction just about ourselves, the EBI team that is here mainly, and then I will talk about motivation and overall objectives of MetabolomicsHub consortium that have been introduced by Tom. Then I will explain the concept. I will explain the data workflow, how it is today, and I will just finish with some conclusions. Okay, so this is not the consortium as a whole. It's just I thought that it was a good idea to include a slide about us. So the Metabolites team is based at the European Bioinformatics Institute, which is very close to Cambridge in the UK. And EBI contains probably the largest collection of biomedical resources and data resources. Ensemble, PDB, Uniprot is there split in different kind of, I need to understand how this works. No, okay, that doesn't work like that. So anyway, I can do it from here, I think. Well, you have to trust me, it's here. So we are based in the chemicals, molecules, and drug discovery part of the resources, and we are responsible for the metabolites resource. So, of course, the main objective of all this consortium as a whole is to make metabolomics, but also related data, lipidomics, exposomics fair, and fair means fundable, accessible, interoperable, and reusable. And we are going to be covering all the acronyms behind FAIR in this presentation, but the first ones are going to be fundable and accessible. And Metabolites is the data resource for metabolomics data in the EBI. It was started approximately in 2010, 2011 by Chris Steinbeck at the time. This is the new website, this is the URL, and is a generalistic data repository that stores all types of metabolomics, lipidomics, and chemical profiling experiments. So this is just a short introduction about ourselves, because here really the point is to announce to tell you that we are not working in isolation anymore. This was a very nice development in the last couple of years, year and a half, that we were started working as a consortium, the MetabolomicsHub consortium, which, the objective is to have international data exchange as first standards for metabolomics. And the three funding resources are all represented here. But Yasin is not here because he missed his flight. But anyway, we are all here. Metabolites, Metabolomics Workbench, and GMPS-Massive. So as Tom said, these are really the main objectives of the consortium, really to standardize data practices in the metabolomics and related fields. Again, lipidomics, exposomics, chemical profiling. So for instance, things that are clear in other fields already for a long time. What is a data set in a public data resource for metabolomics? Which components, which data types, which metadata should it contain? Also, the idea is to have a common voice, the resources, to communicate with journals and funders in the context of open practices. So very often these days, the journals don't really know what to require to metabolomics researchers. What is the data that should be made available in these kind of resources? It's not standardized. We are working on that. And really the overarching objective is really to generalize the availability of metabolomics data in the public domain. Really, metabolomics as an omics approach is really behind other omics, transcriptomics, proteomics in the context of these open data practices, and we need to change that. And really the last objective is to promote or increase data reuse for the benefit of the field. Okay, so these are as I mentioned before, the motivation and the main objective, and now I'm going to explain the overall concept. Okay, so we started working on this on January 2025, so it's really now more or less 1.5 years ago. It was a project funded by CSRI BioHub. Again, the objective here is to implement a standard data submission and dissemination practices between the main metabolomics repositories, metabolites, GMPS-Massive, and Metabolomics Workbench. So this is a completely open framework. That means that we got the original funding, and it's the three of us working right now, but as I will highlight later, it's open. This framework will enable any other kind of resource in the future to join the consortium, and it was developed like that from the beginning. So resources like Metabobank, MBPost, KMAP in Korea, the Indian new metabolomics repository, all of them will be able to join if they want MetabolomicsHub in the future. It's also important to highlight that at the moment we are going to handle open data, fully open data. So at this point, we are not handling human-sensitive data because this is much more complex. It doesn't mean that we will not try to do this in the future, but the logical starting point is to start with open data. So we are not dealing here with control access data sets. Also, because again, we have to start somewhere, we focus on mass spectrometry data, including LCMS and GCMS, but not specifically imaging mass spectrometry or NMR for now. We know that those techniques are very important, but again, we cannot do everything at the same time, and then we needed to start somewhere. We are starting with LCMS and GCMS. So we'll have common data identifiers from data sets, what we call MHD identifiers, in addition to the original identifiers from the different resources. And we also will have common identifiers for spectral libraries. Then we will have a consortium agreement. That means that if one resource runs out of funding, which can happen in the current climate, especially in the current climate of funding for scientific research, then the others, they will do their best efforts to take the data. That means that the data will not be lost for the community. This is quite common to what happens in other fields. Then we'll have, as I will explain, common submission guidelines and data formats. And it also has a first step to have AI-ready data. And this will be presented later by Cal. We'll already have a portal for all public data sets for you to explore. So how is the data workflow? So the data workflow is basically, let me see if I can, I don't think the pointer works. Anyway, you have to believe me. It starts here in the metabolomics community part. Let's see if I can. No. Yeah, here. Yeah, it worked. 1 second. I managed, I think, somehow, but I don't know now how I did it. Okay. Okay, since that's okay. Anyway, for some reason I'm not able to control. Okay, you have to look at this slide carefully. Okay, so it starts here in the Metabolomics community. So everyone who is in this room, everyone who is in this conference, everyone who is working in the field, then they will perform a data submission to any of the 3 resources at the moment: Metabolites, Metabolomics Workbench, and GMPS-Massive. So these are the 3 resources. In the future, there may be others, but at the moment, these are the 3 resources. Then the data will be private by default. It means that during the manuscript review process, we provide credentials for reviewers and editors to access the data, so the data set will be private. But as soon as the publication is out, as soon as the publication is published, then the data will become publicly available in the original resources, but also all the public data sets will be visible in what we call the MetabolomicsHub portal, okay? So this will be a portal where you will be able, it's already there, where you will be able to search for data sets that are stored in any of the 3 resources. It will be a common portal for all the MetabolomicsHub partners. And then that means that the data will be fully available for the community, and data can be downloaded and reused. So to enable this, we have developed a common data model. So you know, metabolites, we have our own data model. GMPS-Massive, they have their own data model. Metabolomics Workbench, you guys have your own data model. In order to coordinate this, in order to standardize, we have to create a common data model. So that means that no matter which resource you use, the submission interface will be slightly different, but the data that will need to be submitted and the metadata will be basically the same. So the common data model is kind of the minimum common denominator of the data that is provided now through the 3 resources. So the data model is graph-enabled and extensible. It means that you know, everyone can work with it with kind of AI tools. For instance, it makes use of common control vocabularies and ontologies to express the same concepts in the same way. And as I mentioned before, it's initially focused on mass spectrometry data only. So this is how it looks graphically. So I'm not going to get into details here, but it's just to realize that the common data model can be represented as a graph, can be also be represented in other different ways. But I think it has been done in a clever way so that it can be easily reused downstream for instance, AI-related applications. So it contains you know, all the kind of metadata that is required for each data set, and also links to the raw data files, result files, and specific information here. There is no point for me to get into the details here. Also for the more kind of technical people, there are a lot of nice features that have been implemented there. If you have any questions, just you know, ask us during the conference, because we will be very happy to explain all this. We are going to finalize the version 1.0 of the common data model. I think we have this deadline by the end of this month. So, you know, in a few days. And again, to highlight that the main concepts are expressed in the same way. So it means that we make use of control vocabularies and ontologies so that, you know, the information that is provided in Metabolomics Workbench, the information that is provided in metabolites is provided in the same way. So we make use of common ontologies like KEBI for chemical identifiers. Eoin is going to talk about it later, but also PSIMS, the mass spectrometry control vocabulary to express all information that has to do with mass spectrometry, EDAN, and others. And yes, just to highlight that we are doing that. And then we also have submission guidelines. So for every data set that goes into MetabolomicsHub consortium, it will be mandatory to have the raw data, so the original experimental MS data recorded by the instrument, the primary data. Then it will also be mandatory the experimental and technical metadata that is required to fulfill the common data model. And then the process results, at the moment they are optional, but for some resources, they will also be mandatory anyway. So for metabolites, if you want to submit a data set to metabolites, you will need to provide, for instance, the option results. But if you do it with GMPS-MASST, for instance, at the moment it's not mandatory. So we decided in this first version to keep process results optional, raw data is mandatory, and then the experimental and technical metadata required to fulfill the data model is also mandatory. Okay, so again, this is the way the whole workflow works. At the end, once every data set is kind of made public, so because the paper has been published, then all the data sets will be searchable, will be discoverable through the MetabolomicsHub portal. This is the presentation that Carl will give later. This is the URL, so I'm not going to talk too much about it. This is just how it looks at the moment, with just the data sets that are there from the last kind of 12, 13 years, and this is how one of these data sets looks in the portal. But Carl is going to explain that later, okay? So at the moment, what I wanted to highlight is we are still in the portal. There are the data sets that have been stored by metabolites, Metabolomics Workbench, and GMPS since 2011, 2012. All the data sets are already there, but we still don't have a new kind of MHD dataset, MetabolomicsHub dataset. We are planning to do that on September. So on September, we are planning to open this framework for everyone in the community. Again, this is not closed. Everyone in the future, if there is here representatives from other resources, they will be able to join in the future. We have a lot of interest from Japan, MBPOS and Metabobank from Korea that came up resource as well. Okay, so what's coming next? So we are finalizing the common data model version 1. We have a deadline by the 30th of June, but I think we are pretty happy, and this first version will not change. The data model, as any kind of data model in bioinformatics, it needs to change dynamically a bit. So the first version is never the definitive one, because one realizes about possible things that could work better in a slightly different way. So we expect that the data model will change a little bit in the next year or so. It will be invisible for users, but it will need to probably change till we are completely happy with it. So we are also about to get the first invited. So we will invite some people, some friends, to provide the complete now MHD submissions, so MetabolomicsHub submissions, also to test the whole infrastructure. We will have to do further tests and refinements, and we plan to go live on September. So on September, if you submit at least to Metabolize and Metabolomics Workbench, we have now we really want to take the first data sets, MetabolomicsHub data sets from the community from September. So that's basically the timeline that we have. And in addition to data sets, we will implement a data workflow for spectral libraries. And that will include the spectral libraries will be giving an identifier. It will be an MHL identifier. And that means that the spectral libraries you can put this identifier in papers, and then it will be much easier to track reuse, to track which spectral libraries are used the most, and these kind of things. So conclusions, just to finish. We are standardizing open data practices in the metabolomics and related fields, lipidomics and exposomics. Really the overall objective is to generalize the availability of metabolomics data sets and spectral libraries ideally in the public domain. Metabolomics is far behind other omics in this context, so I think it's quite important to achieve this. And then I want to highlight that MetabolomicsHub infrastructure for the resources and guidelines are quite advanced already. So we plan to go live to users in September. It's not a good idea to do these things in the middle of the summer, so that's why we put kind of September. I have explained the submission guidelines, the common data model, the data portal and related infrastructure, so that will be explained by Carl later, and the updated submission systems in the resources. And just to finish, I really want to thank everyone that has been participating in the consortium, and I'm happy to take any questions.
Thank you so much. Very great initiative and great job. So I think it's very helpful for the whole community
I just have a question. You said that you are not planning for now to treat human-sensitive data. Just a curiosity, which kind of data are you talking in particular, like age, sex, BMI, these related data?
So when I mean human-sensitive data, it's data that for whatever legal reason cannot be made openly available. So that may be for legal reasons. It may be because of the general framework, like GDPR in Europe, HIPAA in the US. So there are certain data sets that cannot be made fully open. And there must be different reasons for that.
Yeah.
So this is, of course, quite common in genomics. In other omics, it's less common. But basically, it can be also due to patient consent. So the patient must have consented the use of his samples in the data. But so for this type of data sets, control access infrastructure needs to be developed. We are aware of that, but we need to start with open data because it's kind of much simpler.
Yeah, sure.
So did I answer your question?
Yes, sir. Thank you so much.
Okay.
Juan, excellent concept and project. I think the question that was raised, without the human data, what are we going to do, right? People who study human disease. Without having access to clinical data, it's problematic. None of the data sets really have value beyond the development of technology, standardization, and so on.
Well,
so I think, yeah, we do need, as a community, to work because we are struggling with it. We are part of open science within NIA and within NIH, so we've been living the problems and the issues of making data public along with access to human data in some controlled ways. So I think we need to continue the dialogue. Yes, how do people access, and does it have to all reside at your end, or can we link it back, right, to repositories that do house clinical data?
So, yeah. So as I said, I mean, this is a very well-known problem, so you are aware of that. So this framework, what I can say is that it would be perfectly extendable to, to other resources, and they could be resources that are control access. Of course, the metadata model, then it will need to change slightly because there are kind of there are kind of elements that in the data model that would only be applicable to control access data sets. But this is a very, as you know, this is a more part of a discussion. This is a kind of complex model, complex problem, because for genomics sequencing data, this is kind of well established, but for other omics, all the other omics have the same problem. So the funders need to realize that this is something that is required. What I can say, again, from our perspective, is that we have done everything in a way that this is perfectly extendable to control access resources. Yeah. But at the moment we don't have that capability.
We are building bridges also. If there is clinical data that can be accessed, right, you can build a bridge so people can download the biochemical data and be able to get the clinical. The other question, are people willing to deposit raw data, spectral data? Is that uniform now across, that anyone who wants to make data public? And I don't know how many success stories do we have for UCSD, for Fahy. Yeah, do we have good success stories in people reusing the data in metabolomics and making a real impact above and beyond the originators? Can you comment on your experience? We should track. We should track. Are we making an impact? Yes.
So we have. I mean, we can sit together. We have done a study in Metabolize to realize how our data sets are reused. It's kind of the first version. It's not kind of we could do much more than that. And in the last year or so, there have been nice papers about the reuse of public data sets for AI purposes in the community. I don't know if we have prepared some slides from that, but I mean, we could. I have the slides, so I could kind of present them later if there is the time. But it's true, the documentation of how data reuse is done by others is actually quite key for the field as a whole. No more questions? Okay.
Okay, next we'll get on to a favorite topic of the community, metabolite identifiers and names by Eoin. You guys just like to give us the names, and then we expect to know what this name means and what structure annotations. So Eoin will talk you through this.
All right, thanks, Tom. I'm Eoin Fahy from UC San Diego, so we're going to discuss compound identifiers, something we've dealt with in the Metabolomics workbench, and we want to use this, obviously, for the MetabolomicsHub. So as you know, there's a huge amount of metabolite name heterogeneity. Unlike genomics and proteomics, where you have a standardized system of gene symbols and identifiers, there's no such compliance in the world of metabolomics. It's basically everybody from themselves. As you can see from this slide, you have numerous synonyms for the same metabolite, things like two-part names. People put on adducts, isotopic information, user-specific information like A and B, and various suffixes. And this is coming from a lot of proprietary MS software, different open source software, and a huge number of customized in-house workflows. So when we started Metabolomics Workbench back in 2013, we quickly realized this is a huge problem because everybody had their own system, which works great in-house, but when you go out in the real world and compare and contrast, it's a bit of a disaster. So a few years ago, we developed this RefMet standardized system, which we published on. We have a whole section of it on the workbench, and we pipeline all our studies inputted to the workbench through this, and you can obviously harmonize your own data too through the user interfaces. So why do we need it? Huge diversity of reporting names in metabolomics, and we really want to be able to compare and contrast data across different experiments and studies and link those to pathways and other system biology objectives. So we implemented this by taking the actual metabolite names we got through the submissions. So it's an analytical chemistry-centric database because it's based on real data coming in. We've have about 4,800 studies so far in the workbench, over 1.2 million total named metabolites. So we've generated this as time went on over the last 10, 12 years. So what goes on? This is a typical example of the heterogeneity for the same names. So this is what RefMet does. It harmonizes all those names into one simple, commonly used name to basically pull all of these data metabolite names together. And we have a user interface where one can go to the website, you put in your own list of metabolite names, and it will generate the RefMet versions if possible. It's about 85% effective. Why is it not 100% effective? We just get so many names, we don't have the manpower to actually handle every single metabolite name we've ever seen. The second reason is we get some studies where the annotations are, let's say, a bit dubious, and they would have names as long as your arm with all kinds of chiral designations, so you wonder whether these data are really good. So we stick with the more solid studies in terms of adding to RefMet. A very important point here is the levels of annotation. Level 1 is where you have a complete structure. So this would be typically from targeted analyses, where the regiochemistry and the chirality and the double bond geometry is known. The second is the regiochemistry level, where the regiochemistry is known. So very importantly, the top two, you can have PubChem identifiers, KB identifiers, SMILE strings, MOL files for those two. Now the next two, level 3, a molecular species level. So for example, in the world of lipidomics, if you have a phospholipid where the chains are known, but not the SN position or the double bond information. So you cannot draw a structure for these. They do not have PubChem IDs, so that's very important to realize. And often we get mixtures, like people have a hard time distinguishing citric and isocitric acid, leucine, isoleucine, and so on. And then at the lowest level, the species level, where, for example, the total number of carbons and double bonds is known for a particular head group, let's say they're phosphatidylcholine or a ceramide and so on and so forth. All of these are in RefMet, but very importantly, only the top 2 can be tracked to a structure. The other thing that RefMet does is classify all metabolites. So for lipids, we use the lipid maps classification system. It's a well-established system that's been around for about 20 years. For non-lipids, we use various tools. We have classifier developed at the University of Alberta. We have MP classifier developed by Dorstein's group at UC San Diego. And we've also been working on our own custom deep learning algorithm composed of a RefMet training set. It still needs curation before it gets in the RefMet database, and you can see the numbers of superclasses, main classes, and subclasses, so it's a 3-part hierarchy. What's in RefMet? The ones in green are the lipid superclasses. Some very important other classes are the organic acids, which includes amino acids and peptides. So there's a ton of those organic heterocyclic compounds. Other very important classes are alkaloids in the natural products arena, nucleic acids, carbohydrates, and a number of others. And this table is listed with the most commonly encountered species at the top. So we get huge numbers of lipids, for example. And this is another representation of the breakdown of different superclasses. Lipids comprise a huge piece of the pie because there's so much variability in the acyl chain for things like phospholipids, glycerolipids, sphingolipids, and so on. Amino acids are very important, nucleic acids, alkaloids, carbohydrates, and many other classes. This is for Metabolomics Workbench, but I think metabolites breakdown is pretty similar to this as well. Having a classification, we can give people a lot of more information about the subclasses. So for example, if you're looking for acridones, phenazines, what are those structures? Well, you can easily find it here in this classification window to look at the individual structures for the different subclasses. Very importantly, RefMet links to a number of identifiers. For every single molecule, we have the exact mass and formula. For the ones in level 1 and 2, the structures, we have Inchiki, SMILES, PubChem, KEBI, and KEG. And we have a related metabolite database that has many other important ones like lipid maps, HMDB, links to MassBank EU for the actual spectra for that metabolite, and many, many others. So if you look at a RefMet page for one molecule, this is example cholic acid, it's broken down into different levels. So at the top level, you have the RefMet name, ID, systematic name, links to the synonyms, exact mass. The second section for exact structure would be mol file, Inchiki, SMILE strings. Then the next one is the classification. Very important section next is distribution. I'll talk about that in the next slide. And then all the cross-references to the different identifiers, PubChem, lipid maps, KEBI, KEG, and so on. And finally, links to the spectra for that individual compound, NMR and mass spectra at MassBank EU, if they exist. So one really important component of this kind of harmonized annotation is you can look at the distribution of a particular metabolite. What species is it found in? Now we're talking about thousands of studies now, so we have a lot of data. What species is it found in? What's the tissue distribution? Is it found in blood, urine, whatever? What chromatography was used to find that metabolite? Is it found in mass spec or NMR? And finally, NMR metabolite studies that report that molecule. So for something like cholic acid, it's found in very large numbers of studies where you can go and actually look at the data. Where are the standards for that metabolite, and so on and so forth. This is an example of a RefMet page for a partial structure. Now, the structure is not known, so there's no information on mol files, smiles, et cetera, but there is information, again, for the distribution. Where has that molecule been reported? What species, what sample source, what platform, what chromatography system, and so on and so forth. So now I want to switch a little to what is going to be done at the MetabolomicsHub for these identifiers. So this is Jonathan Hunter's work. He's at EBI. He can't be here today, so this is his slide. So he's developed a Python app, a Dockerized Python app, to basically take in identifiers. So that the input data will be coming from the Metabolomics workbench, GMPS, and metabolites files, as Juan Antonio talked about, in this new system. So that'll be the input. The output will be that those files enriched with the identifiers like KB, RefMet name, and so on and so forth. So basically the philosophy is to have a pre-constructed in-house database of, so for example, all the PubChem synonyms are stored in a SQLite database for very rapid access. It accesses the RefMet API too, and many other databases like HMDB locally. So it's a very high throughput. It's traceable. Every metabolite can be traced to its provenance, and it's configurable. And where that stands right now is there's an initial prototype that's complete. So there's a website on Hugging Face that's available privately, and Jonathan is working to get more information and feedback. We're working with GMPS and the workbench to test this out first, but the idea is to improve the informed selection. So if it's making any mistakes, where is it making mistakes, and how can that be improved? It's going to have a built-in classification component in there using classifier and MP classifier and other resources. We're going to do some benchmarking and tests, so we'll do training sets and hold out some sets where we know what the metabolite names and classification is. And hopefully this will be deployed in the near future. So this is the MetabolomicsHub vision of metabolite annotation and harmonization. One final slide, as part of the Metabolomics workbench, we just do something similar with NIH in the Common Fund data ecosystem. So we have all of these NIH-funded omics centers. Many of them are focused on genomics and proteomics. So again, we're doing what the MetabolomicsHub is doing, and basically correlating all of this data for metabolites, genes, proteins, transcripts in this system called a CFDE, which has its own website here. So that, again, will take in the harmonized metabolite data and many other metadata and get the big picture gene biology overview of a particular system. So this is something that you should be aware of too, and this is where the field is going. And finally, I'd like to thank my colleagues at UC San Diego and the San Diego Supercomputer Center, and we're supported by 2 NIH grants, and as well as the CZI Foundation. So thank you very much. Any questions on that?
It's great. The curation is amazing. Your level 3 type of annotation, right? You say you cannot put structures, but you could put partial structures. In fact, some examples you showed at the end, where you have R prime and so on, you still have this core of a structure that tells you that there's a phospholipid. So are you thinking of putting smarts or something similar to having, sort of machine or chemistry form of your knowledge there, to at least say what is that core that makes a flavonoid, makes a phospholipid, and so on. Because also for the classes, that would be tremendously helpful to basically build on that, even though you don't have the full structure.
Yeah, that's a good question. So for things like phospholipids, if you know it's a phosphocholine, you have that core structure and things like that. So yeah, we have the core structures. We just can't represent the entire metabolite annotation in a structure.
But you don't have that in RefMet, or also for the classes, you know, you have so many classes.
Yeah, we have all that class information, yeah.
Okay, thank you.
All right, thank you again.
Okay. So also, I'm going to explain some developments that are going on in the context of data standards that are also being triggered by the MetabolomicsHub consortium. So again, the term fair data is everywhere, so I'm not going to. And basically, before we have been talking about making the data findable and accessible. This is through metabolomics data repositories. In this talk, we are going to I'm going to talk mainly about the interoperable component. That means different things. Also, you know, Eoin before also covered this interoperability by talking about common identifiers for chemicals. In this case, I'm going to be focusing on common data formats. So what does interoperability mean for those of you who don't know? Basically, it means the use of open, or basically how can interoperability be improved? It means use of open data standards so that different tools, like software tools and data resources, can work with the same data formats. So it means, you know, data formats in mass spectrometry and MR are really very complex, so the idea is to have common standard data formats that everyone can work with them. And then also the use of control vocabulary or ontologies so that the same concepts can be represented in the same way. And, you know, one of these components is what Eoin included in his talk before about chemical identifiers. So I always, when I talk about data standards, I always like to include this slide, which, you know, just to realize everybody that the standards are needed in real life. They are needed all the time, and they make our life much easier. So this is just an example from the, you know, socket adapters. And of course, in this case, only a small number of different kind of versions of socket adapters are available. So I think there are like 3 or 4. And it's just to highlight that really with a small number of standards, like in this case 3 or 4, maybe we can kind of, with a small number of converters, kind of thing here, because we all travel now these days with this kind of socket that can really work with any of these. Then this, you know, life becomes much easier. So the Metabolomics Standards Initiative, it was initiated by the pioneers in the field in the mid-2000s, end of the 2000s, and in 2007, for instance, reporting standards for metabolomics analysis. And then in 2012 is when the different public metabolomics databases and repositories were started. This is a super valuable work, reporting standards and community-based MSI templates. But the focus of the Metabolomics Standards Initiative was never put on data formats, okay? So the MSI has developed a lot of very valuable guidelines over the years, but the focus was never data standards. However, in proteomics, kind of things develop a little bit different. At the beginning, there was a lot of focus on guidelines, but immediately that focus shifted towards open data formats. So the UPO Proteomics Standards Initiative develops open data standards originally for proteomics, both data representation and annotation, different working groups. Important thing here started in 2002, so next year it will be 25 years old. And what we have managed to do in recent years is to really merge or bring the proteomics and the metabolomics communities closer together. So it's really an increased interaction of people from the metabolomics field working in the development of open data standards. And there is now in the PSI a metabolomics working group. And the PSI has developed a lot of data standards over the years. Many of them are also used in metabolomics and lipidomics. I'm not going to go through a very extensive review. There are the formats that we could say that are used as input formats, for tools, and there are the other kind of group of formats that are used as output formats. And then here in the middle, we have the control vocabularies and ontologies, the formats for metadata, and then the other types of standards. So one thing that, so these ones that are highlighted in red are standards that have been developed from the beginning with metabolomics in mind, or that can be extended or have been extended from only cover proteomics to also metabolomics. So open format that is a success. It was developed, in 2011 originally, and is used by all disciplines working with mass spec data is MCML. So MCML is a data format for the storage and exchange of mass spectrometry output files. It was developed with full participation of academic researchers, hardware and software vendors. And, well, the version 1 was already released in June 2008, so a long time. But the definitive version was, the 1.1 was released in 2011. And now most people work with MCML and no longer MCXML. So we all work with MCML, I'm sure, if you are working with mass spec data. There are conversion tools from raw files into MCML. The most famous one is ProteWizard, but also there is now Thermo raw file parser and others. And again, MCML is heavily used in other. It was originally developed for proteomics, but it is used by any kind of field that takes mass spectrometry as their main analytical technique. There is another open format coming. It's called MCPIC. There is a publication that tries to explain the concept that was published last year in Journal of Protein Research. And the problem that there is now with MCML is that with the new instruments, the files are becoming incredibly big and more difficult to handle. So there is a group of people that are working in developing this format. At the moment, it's just kind of a prototype. This will take some time to complete. But the idea is to have a new data format for mass spec related data that takes into account kind of modern kind of data formats like Parquet and things like that. And the idea is that at some point in the near future, NCPIC will be there, especially to handle very, very large data sets. And that's ongoing, and I'm not going to talk more about it. But there is, so this is for the input data. There is one kind of need in metabolomics, that is the availability of a standard data format for reporting the results of your study, of your experiment. And this is what we call NCTAB-M. So it's a tab delimited file that has 4 different sections: metadata, the summary level, which are kind of the results are reported at the end, the MS feature and quantification level, and the identification evidence level. You can see it as a kind of different kind of level of evidences in the same file. It's a tab delimited file, and it's now supported or started to be supported by many tools like Lipid Data Analyzer, Lipid Explorer, MSDial, NCMine, GMPS, and MetaboAnalysis. This is work in progress, okay? The format is there. Originally was finished in 2019, but it was not used for different reasons, and we are kind of finalizing it now and putting it in production in collaboration with some of the developers of the most used tools in the field. So this work is, actually the credit goes mainly to Niels Hoffman and Philippine Loyal. They are doing here, like, I don't know, 90% of the work. So we are trying to help, but kind of the credit is theirs, especially for Niels. There is a working group meeting regularly, a call every 2 weeks. If someone is here interested in joining these calls and understanding how this is going, because you want to really have a format for your tool, talk to us and we'll tell you how you can kind of join this working group. They're having different hackathons, and then we have the PSI meeting in Rome the first week of May. Quite a number of people were there, and there were really a lot of advances there. So the specification document, the version 2.1, is really underway. It's a complex format because it needs to, handle many different situations. So it needs to handle, everyone has their own kind of experiment, a different way in which the data is generated, and then the format needs to be able to handle all those cases. I'm not going to get into the details about this. just have to highlight that apart from the data format itself, there is an ecosystem of tools, open source tools, like they are being developed in parallel. So, the data format is kind of the way the information is serialized, is stored, but then you cannot do anything without having the tools for people to use this format. So, as I said, it's MCTAP-M version 2.1. There are different kind of profiles. Anyway, and it's being finalized at the moment. I'm not going to, again, talk too much about the details. Niels Hoffman is the person to talk to if you are interested. If you want to join these efforts, please let us know. But I really want to highlight that this is really a key development from the field. We need one format that everyone can read, that basically the results, what you want to report as an output of your experiment, it is easy to interpret. And this is what we are trying to do. The other format or the other kind of standard that MetabolomicsHub group is promoting is the use of universal spectrum identifiers. So what are universal spectrum identifiers? Again, this is a concept that was developed originally for proteomics. It's a very easy kind of concept. You have a string. This is the string. Sorry, it's a bit low on the slide. But you have a string that you can create based on the dataset identifier, and on the MS-RAN identifier, or the scan identifier. And using this string, you should be able to go to the internet. You should be able to go to any of the MetabolomicsHub resources and see that spectrum in the public domain. This is what happens today in proteomics. Proteomics works really, really well. And in metabolomics, it was pioneered by the GMPS group from Peter Dorrestein, but it's still kind of, they did it in a way that, it was just them. It was not kind of in a standard way that we all agreed how to do it. So we are kind of developing the guidelines to develop formally the USIs for metabolomics. Is what is basically ongoing. Again, widely implemented in the GMPS ecosystem, but it is also, it is possible to access some spectra and other resources there, but no formal specification has been developed, and we need to standardize how we do this also in metabolites or in metabolomics workbench in the future. So this is what we are trying to do. So the work is ongoing, and this will be moving forward, and hopefully next year, so every resource will be able to implement universal spectrum identifiers. Again, what it allows is to one string. You go to the internet, you go to any of the resources, and you can see one particular spectrum supporting one compound annotation. Okay, and just to finalize, I think, just to highlight again the importance of the use of control vocabularies or ontology, so that the same concepts can be represented in the same way across software and data resources. And this is what we are doing also in the context of the PSI MS ontology, the chemical identifiers that were highlighted before by Eoin. But we also want to standardize better the capture of LC related information and columns, and we are working with Michael, who's here in the audience, on to try to standardize that as well. An important thing, again, we will not do this only in metabolites or in metabolomics workbench or in GMPS. We'll do it all together. And then we will be able to, everyone in the community will do it in the same way. We also want to align with other platforms, standards and community initiatives like Protocols.io, the lipidomics reporting checklist, and again, RefMet. And just to finish my talk, always that I talk about data standards, and this is a topic where I have worked a lot over the years. I always like to finish with this slide. I don't know if you know this website, but it's quite nice to get kind of cartoons for presentations. And this is really what we want to avoid. We want to avoid that bioinformaticians have nightmares about how to handle a lot of data formats from very different tools, and hopefully we'll be able to improve the situation. And yeah, thank you very much, especially again to Niels and Philippine, because they are really the drivers of the NCTAP-M format, and also to Yasin and Mingwan for their work on the USI universal spectrum identifiers. And yeah, I'm happy to take any questions. How many people here in the audience are kind of, bioinformaticians, so people that are not mainly wet lab? Don't be shy. It's not. I can also raise my hand. So do you have any, I don't know, we need to open the dialogue here because it seems that people are very shy. So maybe okay, Rima?
Maybe for the discussion,
Yes.
to change practice is difficult. To ask a scientist who has limited resources to adopt new things and, with their data is demanding.
And very slow.
And need to be funded. So I think a big pitch for us as a community is to ensure that every grant that is funded has a companion, since we have to make data public and it's mandatory now in many NIH institutes, right? To be able to ask for the right funding to enable this demanding work is critical. Otherwise, I see the difficulty. Maybe we can discuss.
I completely agree with that. So, okay, so in the bioinformaticians that are here in the audience, have you used any, I guess you have used MCML, I guess? Have you used any kind of other format from the ones that have been highlighted before? Don, you need to pass the microphone and then make people talk, because otherwise people are very shy.
Some people nodded. You have used MCML?
Oh, yeah, sure. I use MCML and I don't know what to say now.
Thank you for all the great presentations. As somebody coming from a computing background, one of the issues that I see is reproducibility. So for example, we have taken some Metabolomics published studies and we tried to recreate exactly the same workflow. We could not, with the information published within the journal, reproduce, go from raw data to the results. Then we contacted the authors. And we said, we're stuck here, and we don't know why the results are not consistent. They could not reproduce their own data. So, you had talked about standardizing things, right? But we still don't have a way of standardizing.
Yep. Okay.
And hang on. There's a standardizing, right, for reproducibility issues. So when a tool is run, I don't know why the tool is not outputting how the tool was run, right? So this is on the people writing the tools. They need to be saying, here's the answer, but here's how the tool was run for reproducibility purposes. There's also issues with, Rima was always talking about having your data AI ready. If we're moving into the age of agentic AI and you want your AI to run your tools, all of them, from raw data to figures that you could put in the paper, you really need to have that very well documented in a structured way, again, for an agentic AI to train on that data and also to reproduce that data and to run new data. So.
Okay. Yeah, I would say to that there are different levels of reproducibility. The reproducibility is not only an issue of metabolomics. Maybe metabolomics is a little bit more, I don't know, things are more difficult to reproduce, and one key factor here is the lack of availability of the spectral libraries. That's an important factor there. This problem is not in other omics. About the parameters for the tools to run, completely agree. I think that there are kind of initiatives more and more from journals and from to specify every single data about how the tool was run, the version, the parameters. So in terms of the tools, I don't see this as a big problem in the sense that it can be, the technology will improve all the time. The Jupyter notebooks and all these kind of things is maybe more fundamental in the context of metabolomics, and also based on my previous experience, the lack of spectral libraries that are publicly available, and then this makes reproducibility even harder than from other omics.
So we're not talking about reproducibility with spectral libraries. You could always say, this is the spectral library that I used, and this is the version that I used, right? So that's documenting how you created the data. So it's really about documenting how the data was created more than anything else.
Yeah, that's a huge issue with things like pre-processing.
Yeah.
For example, peak picking. I mean,
And how much manual curation
Yeah.
goes into
Yeah.
the final assignment and pre-processing, normalization. So many steps,
Yeah.
many steps.
but why is it not documented?
Maybe we
It should be. It should be, of
It should
course.
be documented to the level that you could recreate exactly the same thing. So when I'm looking at a MGF file, I should know the provenance of where that came from and what process led from the raw data to that
No,
MGF
you're
file.
dead right. I wouldn't be able to reproduce anybody's data. Nobody. I've been doing this for 40 years. No way.
Okay, can I
For those reasons.
But this is an issue of the standards, right? So I feel like the standards are doing a great job and they've come a long ways, but we still need to do so much more in order to get to the point where Eoin was saying, you need to get it to where I can reproduce everything from the raw data to the peak that I'm looking at.
In 2007, we put a minimal standards initiative as a metabolomic society and community. It was a beginning of being light, right? We didn't want to weigh heavy. We said, listen, let's agree on a minimal list of things that we can do as a community. Maybe it's time to revisit. You guys, it's...
More in terms of the peak picking and
Technical details.
So, I really worry when I see these standards up and saying, oh, this is optional. 10 years ago it was optional. 10 years later, we really need to make these kinds of things mandatory. Otherwise, the data is really underutilized and undervalued because we're not able to reproduce it and reprocess it.
Yeah, it's definitely a black box situation right now in terms of producing data.
I can just maybe mention on the MZM side, we are thinking about this. This is discussion that we're trying to say because I think having the results and the protocol on how you got the results together would be good. But I think we need more people to also have this discussion because there's different ways to do data analysis, whether you do it with graphical user interface or script-based. Script-based is easier to report. We could link to some Markdown or Jupyter Notebook script. That's easy, but some graphical user interface would be a bit more complicated to report. So I think this is discussion that we need to have on how to report. And then the mandatory part, this is on the repository side to see how to implement this, I guess.
And I didn't realize that you were here.
Hi.
So otherwise, you should have been given this presentation.
You did a great job.
When we use MZMine, for example, I'm a huge user of MZMine, and we can report all the parameters with MZBatch file. So I don't know if Compound Discover, for example, can generate a file like that, but if we have already a tool, that generates us these parameters, like I can just update the MZBatch file, and you can open MZMine, and you know all the parameters I used in my data. And why don't we use this as standard? Like if Compound Discover updates this file, like gives you this file of parameters, okay, you can update it. If I don't know MS Dial, I don't use it, but if it does, you have to put it there. Should everyone know the parameters? No? And if you don't use a software that gives you the parameters, you shouldn't be able to put it on the cloud, in the metabolites and stuff. I don't know what you think about it, but it would be easier if you use softwares that gives you the parameters of each process you do with your raw data.
So, okay, so we are talking about different topics here. So I think that a resource like metabolites or metabolous workbench or GMPS, they have different missions. One mission is to store data that have been produced by others and to make it as reusable as possible. Maybe people have to work a little bit harder to try to reproduce what others have done. And this is kind of, is not, again, not only a problem, this kind of discussion is not only a problem in metabolomics. But one thing is to make the data available and have the possibility to reproduce. And another kind of level is to make the data or make the whole workflow fully reproducible. These are kind of two different levels of complexity. I agree that we need to move more and more towards this kind of enabling the full kind of reproducibility. But again, there are many issues that we are aware here. There are tools that are not open access. There are spectral libraries that are not made available. There are kind of tools that are not perfect in reporting the parameters. But still, I think it's very valuable to have data that is there and that, in the future, when the situation improves, maybe they can be reused much more successfully. And also reproducibility is only one aspect. As we know, in mass spec in general, the software evolves all the time. So again, I have, because of my background, I have a lot of experience in reusing data for proteomics that were kind of created with a different purpose to what they have been reused at the end. And this is something that as a field also we should be kind of looking forward to have in the future. So again, one thing is to have the data available, fair accessibility, and one other complete different level of complexity is to make the data fully reproducible. It's kind of, or the workflow fully reproducible. Agree, or would be ideal to have this fully reproducibility, but it is not possible to, it is only possible to do it in certain scenarios.
Over the past 2 years, we have witnessed changes within the NIH and the main funding agencies in how we can work globally as communities, right? And how can money flow from our grants to European partners and so on, right? I think maybe it's being resolved or solutions are being provided. Does this impact what you are doing here as US, Europe, global, Asia? Has the change in NIH rules impacted your ability to exchange, even exchange of data? We have to report anything that goes outside the US. How does this impact such an important initiative?
So for open data, because again, this is open data, at the moment, my understanding is that there are no restrictions. I think the NIH hasn't really restricted that the data generated in the US needs to stay in the US. This may be the case for sensitive data, but not for data that can go from, and in the EBI, we still have a lot of data coming from the US that goes there. But it has impacted in all different ways in terms of funding, of course, but also in terms of collaboration. So my understanding, again, I don't work for the NIH, is that now if you want to publish, you have NIH funding, and you want to publish a paper with collaborators that are from outside of the US, you need to ask now for a kind of permission to do it, or at least you need to report that you want to do it. It's not a problem to do it with people like us in Europe, but maybe it may be a problem to do it if you want to publish, I don't know, with someone from, I don't know, just to put something from China, just to put an example. So that's something that is kind of, but again, I'm not NIH funded. And of course, all these problems affect the global scientific community. And there are resources like UniProt, for instance. UniProt is very well known. It has a lot of funding from NIH, and there are a lot of problems ongoing right now because, of course, UniProt, the main components of UniProt are based in Europe, not in the US, and then there are all these problems related with funding because of the NIH new rules. We could talk about this for a long time, but yes?
Okay, on reproducibility. So you're perfectly right. That stuff is going on. Ming is trying to reprocess all of the data that has been accumulated in all of the databases, so that starting from a public available library, which is versioned, you run a version type of software, which is publicly available, and then you get one result, which is going to be conditioned to that. And all of the studies will be reprocessed. I'm trying to convince these guys to do the same. Maybe with other type of software, MZMine is already providing you the fact that, you have the parameters, you run your batch, you get the same results with the same versions as around the clock. We have R that is basically providing, not R, the people doing this in our entire ecosystem, which is really clean. You have clear parameterization, you have clear version, you get always the same results. So we are not yet there, but I think people are working to get there where the processing also is transparent. We also did our own, doesn't matter, but the idea is really that you get with the same data, with the library, which is public, that's why we're pushing everything out, you get the same results. And can we discuss about algorithms and so on? It's going on, it might take 1 or 2 years, but I think the community is trying to fill that gap that you mentioned.
Just to highlight, when I was talking before about reproducibility, I was only talking about the reproducibility of what people report in papers. That's what is difficult to reproduce. But what Nicola said, it's not that difficult to reproduce if you do the reprocessing yourself. If you have the input files, you know the software that you have used, then you can document perfectly the parameters that you have used, and then you also have the results at the end. So if you do that kind of using the same kind of framework and you do it yourself, then it's very easy to reproduce. You have the input files, you have the software and the parameters, everything is open and transparent, and you have the library and you have the final results. What is now difficult to reproduce is what other people are doing in their own studies in papers. That's what I was talking before, that is difficult to reproduce.
So I think my concern is, I really love what you guys are doing, and I'm wondering if there needs to be another working group on workflow reproducibility, because that is really important, and coming up with a standard on how to represent it, how we feed it into AI. You're laughing. Is this going on as we speak? But if you guys are not doing it, I don't know who's going to do it, but we need somebody from the community, a working group, really start working on that.
Yeah, maybe just one point to add. We had a slide of what other community activities we want to support, and maybe this. Like the MZMine is a great example. It's only new because of the wizard that they've implemented. But there are other things out there. Galaxy, for example, the Galaxy platform, you can export your whole workflow. You can share it. They can plug it in and do it again. So maybe we can coalesce around some initiatives like this that we at the hub we can support. Say, give it to us, and if you give it to us, we can enable your data to be more reusable and more citations, more collaboration.
But it's really a kind of a cycle, right? So some of the tools are starting to better document for reproducibility, but also for the raw data to end product, we really need to be developing a standard. And that's not going to happen by any one individual MZMine, because they're only part of the big chain. And there's no company or entity right now that's doing end-to-end, right? So this is more on us as the community to really start thinking about how to put standards going for the whole workflow. It's done. Right now.
So maybe one comment directly to this. I think there's a big effort on the NF Core platform where people are trying to upload the different parts of the workflow, which you can then patch together. You can put it in a GUI. It is then reproducible, but we need to put more work on to have the different parts of the workflow represented, because at the moment it's still very minimal on what is there. This looks very different for transcriptome or proteome, but I think that could be a good platform to actually work with this just as a response to that one. But what I actually wanted to ask is, for the data formats you're suggesting, is there a place where groups can provide the identification? Because I understand that, everyone can use their own tools to then look at the spectra to get some identities for the potential metabolites. But there is a lot of different ways people are generating the data. There's a lot of semi-targeted platforms where they know more about the identity, which we don't know if we just have the raw file. So what I personally think would be very valuable for the community is that people who have that additional information to submit it with the spectral data. Because if I now download the data and I just use a tool to get identification, it's always going to be worse than having the info from the actual curators about their library. So is there a way this can be added into that?
I mean, at the moment, as you know, for instance, also Metabolomics Workbench, you have your own format, NW-tab. So for cases where it is not possible to use NC-tab-N in the future, it will still be possible to use the map file or the MWTAB file.
No, what we really need is provenance from a particular metabolite in a particular study back to the raw data.
Yes.
Which scan is it?
Sorry, what?
Which scan number is it?
That's supported in
Which
NCTAB
10 MS is it?
So
'Cause
NCTAB
that would
N-
really help.
Sorry, I forgot to mention that. NCTAB N, one of the key features is it supports a linking to the original raw spectra in the file.
But we never get this data from any study, provenance back to the individual spectrum in the raw files.
If MCTAB M becomes popular and is supported by tools like NCMine, then yes, because then it will, for the tool, it is possible to create that link between the compound annotation and the spectrum.
That's all we
need. So again, this is in
right.
proteomics is is the common theme for 15 years at least. So in metabolomics it should be possible as well. It's just one of the key features of MCTAB M is to enable that linking between the scan and the compound annotation. If that information is not encoded in some kind of data format, then it's impossible for the data resources like us to retrieve that information. So that's why this is one of the key features of NCTAB M. Sorry.
That's nice when everybody is giving an input about how reproduce and everything. But I would like to ask the most basic question: the conversion of the data to the fair system, like the fair access. Because when I was working with Brookers.d files and converting it to the MZML, most of the information was lost. And I was working with
that's
Ming.
very true.
I was working with Ming closely to generate a spectral library. And what I was facing, the spectral information is messed up, lost. But when I'm working with the .d file itself, it works. But for this OpenAI or Access, whatever software you want to make fair policies and all, it doesn't work with the .d file. You have to convert it to some format which is well accessible through the community. So I'm thinking and wondering if you guys have some suggestion for those specific software which is compatible for Brookers data, which has ion mobility data incorporated in it.
So, I mean, for me at present, the best solution is to have both files. So the D files from Brooker as a directory, we would kind of request to have them, and together with the MCML files. Because if we have the raw data, is when all the rich information is there, and hopefully this situation will improve in the future. And for this, of course, the vendors also have to put some effort on this. Like, for instance, the formats like MCPIC that they have been there. But to try to solve this, which is kind of multifactorial problem, and the vendors really need to, in a way, have the pressure from their clients to enable this kind of functionality, and this is a key point. The vendors, if the clients, as a client, we don't tell the vendors, oh, we really need this from you, they will not do it. So at the moment, the solution is to have, on one hand, always to have the raw data available as well, in addition to the open format.
If I may answer to this, we're pressuring the vendor. We asked if their format of conversion of .d file into MZML will be functional. And they provided some version, which was actually not a very productive one. And they keep on saying that if with the next version, it might be better, the next version, and the next version never arrived. Whereas what me and Ming are planning to do at the moment, we are using the .d file to convert it into the spectral library as from the .d file into the GNPS system. I think that is much more nice, but when it comes to this fair policy and giving it to the AI formatted data, I still think we need to have to address this, because the Teams convert, which is suitable for this, doesn't work.
Yeah, and this is yet another problem of the long list of problems that are there.
Yeah, maybe just to highlight that there is a new initiative, MZPeak, and this is one of their biggest. Yeah, it's still being developed, but this is one of their biggest objectives. Is to improve the metadata around the spectrum. Oh, okay.
It's on something else.
Maybe we can continue unless any last points.
So do you want to say anything else, Nicola, about this? Okay. Okay, so maybe the next one.
So the plan was to have 2 demonstrations, one around structureMASST through Yasin. But because he's not here, I'm just going to quickly give an overview of the tool because I think it's such a super good tool. And it's one of these tools that really shows the value of sharing your data in the public domain. And then the second demonstration will be the MetabolomicsHub portal itself, which we'll get to in just 5 minutes. So there was always the question of what molecules are here in the public domain. And it was always kind of the way to do it was I have my raw MS, and I go and look, and I want to look against it in a library, right? And then these libraries, if they're in-house, they're kind of curated with what instrument, what collision energy, and so forth. And then you find it, and then you report it as that molecule. But actually what Yasin and Peter and Ming do at GMPS is they try to do it the other way around, right? They say, I have my molecule, where is it in the public domain? And when it's in the public domain, what annotations are associated to that data? So, you can look at the organism, you can look at other tissue and autonomy, and then other factors associated to it, like lifestyle factors, even geographical location. So the idea is I have my structure, let's go and find it in the public domain, let's kind of build a new hypothesis, and where is it found, where is it not found? So with structureMASST, it's built upon MASS. So if you're not familiar with what MASS is the mass spectrometry, public mass spectrometry search tool, and it works on the basis of what happens in the genomics world, right? I have a sequence, or I have an MS spectra. Let me go and see wherever it's found in the public domain. So this is what GMPS did. And what they do with MASS now is instead of a spectra, give just a structure. You give it a structure, they go to their library, they convert that structure into a spectrum within the library, and then within the library they go and search against the public domain, and then they bring you back all the annotations with that spectra as well. Because now I think Peter's number when I spoke to him last was like, God knows how many billion of MS spectra that are now in the public domain. They have to be a bit smarter on how they provide that access to the community. So now they have kind of this whole idea of fast, where you can search billions and billions of MS2 spectra in seconds and minutes. And this is done in 2 ways. There's a fast record, which is a pre-computated annotation indexing system. And then there's also the fast, which actually lets you go back to the raw MS and do some substructure searches as well. So here's the URL. And Yasin, again, has a talk on Tuesday afternoon at one of the sessions, so I encourage you to go and listen to his talk. Basically, this is the interface, right? So I go to the website, I have a compound, maybe I even have an identifier for the compound, I give it to structureMASST, sorry. I say what kind of matching do I want to do, exact or similarity, substructure. So here they're using caffeine. And then they go off and they find caffeine in their library, they have their library, they propagate the library into the public domain, and then they can subset it, right? They can say, oh, in the public domain, these are all the analyses that I find to it. Here's the ion modes that are associated to the public MS2 spectra. I can also subset to the adduct and then also to the collision energy. And then, of course, what else you can do with that, you can then start to pin the metadata on top of it as well. So, with MASS, it was simply just the MS2 spectra, but then they went down into different flavors of MASS, like microbe MASS and food MASS, where, that I can tell you that this MS2 spectra was associated with this data set through this sample source, whether it's food or an organism. So then you can start to really interrogate, right? Where's this MS2 spectra found in tissue? Where's it found in the sample type? And where's it found in the species as well? So he did it with here in caffeine, and you can see exactly all the accessions it was found in, and a lot of blood serum, animals, and of course Homo sapiens as well. Yeah, so that's the 5-minute whistle tour. I won't get any more detail, but yeah, it's a super good tool. It's one of these tools that the MetabolomicsHub want to support. So we want to encourage the use of the amount of public data sets in the domain, but then we also want to bridge the tools, bridge the data with the tools and the workflows, right? We want to show the value of public reuse, and this is one great example of why you should share your public data and what you can do when public data is shared at scale.
Okay, good morning, everyone. My name is Callum Martin. I'm a software engineer. I'm a colleague of Tom and Juan's at the EBI. I'm going to be continuing on from some of the things that they're talking about in MetabolomicsHub. I'm going to run through the portal, and if the technology permits, I will give a short demonstration of the portal, but we are not sure if that's actually possible, but we'll find out. Okay, so I'll go through the basics of the portal, the development processes. If allowed, I'll go through a demonstration, and then we can talk about next steps and questions at the end. Okay, so this is a brief overview of the portal. It is available now. It is a centralized web portal that enables cross-repository discovery for all of the data sets made public in the 3 source repositories. It allows you to search between legacy and MS data sets, legacy encompassing all the data sets that were made public prior to the existence of the hub, and MS meaning data sets that are sort of hub native. We don't have any of those yet, but we will from September onwards. It exposes discoverability across studies, samples, assays, files, and compounds. That's just for now. We are developing further aspects of search currently and for the future. We do not offer download capabilities. We redirect to source repositories to further interrogate the data when you've made your searches. The potential future directions will include integration with existing tools such as the software suite GMPS and the query system over a graph database. And it is available now. I'll talk a little bit about how we've designed the search for the portal in tandem with the common data model. So it's a modular multi-stage query pipeline. So it's not a single monolithic search. This is multi-stage because the common data model is complex. It's going to be evolving in response to community feedback. It's going to go through multiple versions. So we wanted to design the search in such a way that allowed you to build complex multi-stage queries, and you could make changes to the underlying model, to the kind of data you're querying without needing to make substantial changes to how the search actually functioned. So it actually exposes a kind of dual search interface. We wanted to provide multiple ways to interrogate the data. So we have facets for general exploration and an advanced composer that allows you to do more precise multi-stage querying. We also have exposed discovery endpoints that enable programmatic access from day one. I'll talk a bit more about those endpoints in a later slide. And from a technical perspective, it is built on Elasticsearch and Postgres on the indexing and database side, and on Fast API Next from the API and UI side. I'll talk a little bit about this advanced search course. So as part of this querying functionality, we wanted to develop a robust sort of set of logic for doing these advanced searches. So we developed this library called Metabolomics Advanced Search Core. This is in use both in MetabolomicsHub and Metabolites. It provides a sort of common, complex, robust logic for doing queries, establishes types, utility methods. I won't talk about it too much. If anyone's interested, please do ask, and we can talk about it later. Okay, so available now also is the search API. So there's quite comprehensive API documentation already for advanced search, including example queries. But we've also made it programmatic and LLM access ready with discovery endpoints. So we have, amongst other things, we have example endpoints that expose real requests, including the request shape, example clause terms, operators. We also expose a fields endpoint, which tells you what the queryable fields are, what their types are, and what the operators you can perform on them are, and the raw mapping. So this exposes every single field that's an Elasticsearch, whether it is queryable or not. The idea being that all of these endpoints put together, all this information, is enough information not just for a human user to get started using advanced search, but also for an LLM user. So we've had some good success internally running blind Claude code sessions, asking it to summarize these endpoints and to do a basic semantic search. And it was successful. We have not tested this too robustly, but the early signs are promising. Okay, if the technology allows, I'll run through a quick demo. So I can see on the big projector, but I can't see it on the screen, so I'm going to do my best to deliver the demo. So this is what you see when you land on the MetabolomicsHub search portal. You are presented with some highlights of the data at the top. So that is a cross-repository breakdown by organism, organism part, and disease. So if I cycle over here, this functionality will be made more sort of sophisticated in the future. This is just the first release version of the hub. You can see the interface that I was talking about on some of the previous slides here. So there are these facets on the left that expose sort of big picture facet information. And there's an advanced search composer down here. So I will just show you. So let's say I want to filter by Homo sapiens. That brings the data down to roughly 2,300 data sets. But say I want to drill in a little bit more. I can click into the advanced composer. I can say I'm looking for organism parts. Okay. And let's say I have some strange requirements. I want urine or serum. Okay, so that's both. There are no data sets that have both, but if you change that to or, you get a healthy amount. And let's say I want to drill down a little bit further. I want to find data sets that report metabolites, citrates, and let's say I am mostly concerned with data sets that were released after a certain point in time. So I will. Okay, there we go. So with all those filters applied here, so with the big picture of the organism being Homo sapiens, and the advanced clauses being, show me data sets with urine or serum that report this metabolite released after this date, you can see here we've got 8 or 6 data sets left over. You can, of course, there's a free text search field here. And if you wanted to share this data set, if you wanted to share this search, sorry, if you thought you wanted someone else to interrogate this or save it for later, you can click this button here, and with any luck, if I hit Command V, hit that, it should interpolate the exact same queries that you just put in. So that link encodes everything into the URL. You'll have the exact same search terms that you just applied. As well as that, you can, as long as this works, you can download your results, whether there's 6 data sets or 6,000, you can download your results by hitting the export button. It'll export all of those to JSON if you want to go ahead and crunch those in your own workflows. And as well as that, there is, so currently we only have legacy data available on the hub. As we mentioned, in September, we'll be starting to get MS native data sets, but there is individual data set views. So this is a workbench study. This is what we call a legacy profile data set. So the information available here is relatively minimal compared to an MS data set. This is a conscious choice because of the 12 or 13-year history of the data sets submitted to the repositories. We wanted to give a kind of homogeneous presentation for legacy data sets that included core information that sort of accounted for the wide variety of completeness and quality of data across those 12 years in repositories. So it includes just basic but important metadata information. The full MS native data sets will include a lot more information to do with files, reported metabolites, but this is what you get when you're looking at some of the older studies. Okay, that's it for the demo. If possible, of course. Oh, yeah, to mention, there is documentation available here. If you click this, the documentation will take you to documentation not just for the search, but for primarily the model itself. So you can interrogate both profiles of graph model. You can go and see the full extent of the quality of data that we expect once we are taking MS datasets in submission. And we encourage you to please go ahead and use both the portal and the documentation now.
Yeah, great. Thanks, Callum. Yeah, so this is a new development. It's a prototype at the moment, and we're now looking to refine it, so we're very open to feedback. You know, are we missing anything? Are things not making sense? And like Cal said, this is for the old data to begin with. In September, we hope to have the new data sets that, you know, have a lot more information that complies to the minimum requirements that the consortium thinks are important now for a data set. Yeah, but very excited that you can come now to one portal and you can see all the data sets across the 3 repositories. hopefully in the future, you come there and there's a data set from Japan, there's a data set from Korea, India, etc. So yeah, and so we have 10 minutes left, so I'm going to open the floor for questions, feedback, comments.
I maybe can quickly start. So I've actually been trying to go on the portal for the last few weeks because I'm super excited by this. I think it's a really, really great initiative, so thanks for that. So one very small comment. So you showed that you can share the link to save essentially the settings, right? Mm-hmm. Maybe it's just me, but could it also be somehow frozen in time also just to enable also reproducibility? Because the link might be that it's not going to give you the same data set back 2 years from now on, right? So if we could have like something a bit more static, just for the reproducibility side of it, could also be nice. Or like some session setting, whatever. That's just a small
That's kind of a good point. I think, so that's not currently something that is enabled. But I think as the hub matures, you could start to think about having versions of indexes. So in that same encoding, you could say, this link was produced with this particular version of the index of data sets. So like say in 2 years' time, you're not hitting current indexing where the model might have changed, there might be data sets that have been added or even removed. You could go back in time to the index in its state of 3 years ago. But no, that's a good idea. We can go with that.
And then you mentioned the legacy data set. How do you have any minimal metadata that you're asking for these legacy data set or not? Because I know like from the one from Massive, for example, some just don't have anything. Are they here or not?
So yeah, Massive, so minimum requirements is super, super minimum. All I need is maybe a title and maybe a contact, so a PI or a submitter. And that's it to get on the data and to be on the, so indexable. But the philosophy is very much that the repositories will, if they have that information, give more than that. So Metabolites, because we will give a lot more. We will give samples, information. Workbench will also give, but still under the same tag legacy. And then if it's there, it's not there. If it's there, not. Very much we're looking forward to the new data sets. Come 2 years down the line, there'll be 6,500 data sets, but there'll be a lot more new MHD, MS ones, like thousands. So they'll be diluted out in the future. And again, this is going to evolve. So, this is version 1.0, version 1.1, 1.2, etc. will come. And hopefully as the community matures, the requirements will also evolve as well. So, maybe we can think about as a community when MZTab-M becomes prevalent, that now becomes a mandatory requirement. We spent a lot of effort on the common data model, try to make it like graph-enabled and future-proofed towards these AI opportunities in the future. So if anyone has any feedback on that, particularly interested. I think there was a discussion earlier around reuse of data and what people are doing with public data. There are now these new foundational models. Thomas Pruskow's group, DreamMS. There's the Princeton, Michael Schneider in Princeton, who do deep Met. So yeah, we're very keen on how we can make public data a driver for AI research in metabolomics. We're a little bit behind with other communities, such as genomics and proteomics. No, you have 5 minutes left, no coffee yet.
I have a quick question about the panReDU otherwise initiative, just how this is overlapping with this, but we can finish. Sorry, just wondering because there's also panReDU that came out a few months ago, and how is this kind of overlapping with that?
So panReDU was basically what so it's a first step of this. So it's a sample level aggregator. So basically it says, any data set in the public domain that has MS2 data files that has at least one annotation, and the one annotation is often organism. Most public data files will tell you what organism they're studying, and then the index will harmonize that across the repositories. That very much continues, but this is really building upon that effort. So all the MetabolomicsHub data sets will be in panReDU, but with much more granularity and more harmonization across the Do I have to start picking on people? Maybe just one last few words is, we'll be around the conference for the rest of the 3-4 days. So yeah, just come by. There's a MetabolomicsHub oral presentation on Tuesday, and then Yasin will, after that, followed by Yasin's presentation on structureMASST and fast records. And then we also have the Metabolites poster as well on the last 2 days. So yeah, come find us and say hello. Thank you.